The next recipient is Professor Michael Buckland from the Brain Mind Research Institute, Royal Prince Alfred Hospital, University of Sydney.
The Brain Foundation and all the generous donors. This is an invaluable award for our research. I'm going to tell you a bit about microvesicles. These are small little membrane-bound packages that are released from cells. Now, they've actually been known about for quite some time, but for many years it was thought that just some blood cells released these little packages, and they contained proteins to help the blood clot, and that was the end of the story. Now, over the last 5 or so years, it's become evident that many different types of cells in the body actually release these microvesicles, and probably more exciting is that they don't only contain proteins, but many cells pack them full of RNA and DNA, which is the information of the cell. And these can be taken up by other cells around them, and that information then can affect the genome of surrounding cells. So we've sort of dubbed it the biological equivalent of tweeting, in that one tumour cell can send out all this information to many hundreds of cells around it and influence the transcriptional landscape and the genomic architecture of all these other cells. We're particularly interested in them in glioblastoma. Glioblastoma is the most common and most malignant of the primary adult brain tumours. It's a death sentence to everyone that gets it, and despite surgery, radiation therapy and chemotherapy, everyone eventually dies of the disease, and the median survival is still only about 12 to 14 months. We think that glioblastomas use these small microvesicles to tell the surrounding landscape of the brain to let them grow, basically to make a fertile soil for the normal brain to allow the tumour cells to grow. So our project is to understand how these glioblastoma microvesicles interact with normal brain cells and change the normal brain architecture. And we hope eventually this will be a novel avenue for therapeutics. So thank you again.